All right, we are live. It's 8:30, May 5th. I'm Richard. I'm co-founder of Escarpment Labs here presenting the continuation of our webinar series. We took a little break last week, a much needed break after our intensive conference, EscarpCon. That was the name we came up with at the time. Really, there was just nothing else better that came up, and I'm glad we did it. We were able to think put together like nine talks in the span of 3 days. Try to bring on some exciting guests like Lars Marius Garshall, and then some of the other researchers that we've been fortunate to work with, like Christopher Krogeris and Caroline Tarawa. Try to put together a lot of awesome content, and then totally burnt ourselves out. Took a week off, regrouped, and put together a plan for some more webinars moving forward. So welcome to sort of the new format for the Escarpment Labs webinar series. We're going to be keeping these presentation style webinars going every Tuesday, where we're going to do a deep dive into some kind of technical educational topic about fermentation and quality control, yeast in general. And then every Thursday, we're going to try to do an interview with the brewers that we are lucky to work with. A lot of these people have, really awesome subject matter expertise in different things, and really excited to bring on some of our brewer friends and talk about beer. And a lot of these people don't always get a chance to talk about the ins and outs and the technical nitty gritty of what they're doing. So I think we're really excited for that, as opposed to just like, how did you get into beer and what's your favorite beer, right? And I think we have some exciting stuff lined up for that. Our next brewer interview is this Thursday. It'll be with Avery Swanson, formerly of Jester King Brewery, now with her own project, Keeping Together, based in Chicago. And we're going to be talking about saisons. We're going to be talking about mixed fermentation beers. A lot of really insightful stuff. She's been doing, working on sort of the top end of brewing for a little while and making some really, really cool stuff, and continuing to push the envelope with yeast and fermentation and all the ingredients that we can use in beer. So really excited for that. We're also going to be back on Tuesday next week, where we're going to be talking about good wild yeast hunting with Jonah Greenbaum Schinder from our lab. Jonah's been doing a lot of work with capturing, testing out wild yeasts for ourselves and for our brewery friends, and really excited to see what Jonah has to say about catching wild yeast. And I know a lot of people were asking about this in our previous webinars as well. So hopefully that's some useful information. So that's it for me. That's my spiel. Today we've got Alex Mitro, from formerly of our lab team, now doing a little bit more of a business development role. Alex has really been spearheading, helping us make the business better, grow in new directions. Really excited to see Alex grow into that role. But for a long time, he was helping us get our lab up and running, get all the SOPs in place. And one of the things that he really specialized in was PCR and setting up ways to make it easy and cheap and repeatable for us, which is, this isn't limited to beer. Everyone has problems with PCR. It's always a pain in the ass. And so really appreciate Alex's effort and insight in helping to make that easier for us. And I hope that he's able to make that easier for you guys as well. And I know, a lot of brewers sort of know what PCR is, but, maybe you're afraid to ask, right? Or maybe you don't know the details that might help make it possible for you to start using it, or to start getting a little more value from the system you already bought. So that's our hope here, is to make that easier. So without further ado, I'm gonna give Alex the floor for this presentation. We're always gonna have a Q&A at the end. So feel free to ask questions during the crowdcast. Feel free to upvote other people's questions as well. And we're gonna do our best to answer all of them after the presentation.
Awesome. Well, thanks, Richard. I know I'm really excited personally for Jonah's talk. I haven't seen it yet, and I help him out a little bit with the wild capture as well. So that would be really great just to see what he's done with it all. So I guess I'll get started. Do you have to share my screen, or do I have to share my screen?
You have to share your screen.
There we go. Welcome to PCR Basics for Brewers. We're Escarpment Labs. Normally we have our shit together, and I apologize for all that. But anyways, we are a Canadian yeast supplier. We offer fresh, locally made, and ready to pitch liquid yeast to craft brewers. We also provide a wide range of services to the brewing industry, including microbiology, quality control, custom yeast and bacteria development, as well as strain banking and in rare cases, wild capture projects. And we also strive to be excellent sources of knowledge, with our company focus being on quality and education, which is sort of why we brought you these webinars. So this is me, Alex. I have a Master's of Biotech from the University of Guelph. My previous role was, as Richard mentioned, to develop and manage the external quality control department, doing a little bit more of a lab-focused role. I enjoy doing that, so now I do that sort of stuff throughout the company. I'm a business development and project management role now. So I really like crispy beers, especially anything crushable or fruited sour. Definitely a big fan of Coors Light, but I'm not a brewer. I've brewed a handful of times and they've gone questionable. So this webinar is fairly condensed. It's pretty technical. I have technical difficulties with computers, so please bear with me. But the goal is to provide an overall background knowledge of the polymerase chain reaction, or PCR, backgrounds and principle, as well as explain to you guys how to set up a PCR reaction, which includes performing a DNA extraction and running gel electrophoresis to detect successful amplification of your experiment. And then lastly, to explain how you can kind of adapt existing equipment you may have purchased, such as the Invisible Sentinel system, to do some in-house testing that doesn't require such expensive kits. So we'll get started. So before we get started, there are some terms that will be used kind of throughout the presentation that I might just throw out. So I thought I should explain them a little bit. So a gene is a segment of DNA that forms a functional unit, and these are hereditary, so they are inherited through your parents. Many genes act as a set of instructions, and these instructions produce proteins. These proteins then perform actions or perform some sort of role in the body or cell. So, for example, STA1 would be your gene. This gene encodes for a protein called glucoamylase, and the glucoamylase will perform additional functions inside of the yeast cells. Replication is when DNA polymerase actually reads the DNA template to synthesize a new strand of DNA, and this occurs in a 5' to 3' direction. I will explain that again in a little bit. And supernatant. So supernatant is when there's liquid above a solid residue. So if you have a yeast slurry and you spin it down, your pellet would be your yeast cells and any sort of debris, and your supernatant would be the remaining liquid without anything in it. So a background. Before you can run a PCR, you do need to do some prep work, which includes extracting and purifying your DNA from your source material, for example, yeast. Develop primers to target the gene of interest that you're looking to find if your yeast has it. So in this case, STA1. But you can search for anything within the genome as long as you have primers specific to that area. And then you need a PCR reaction mixture, also known as a master mix, to incorporate all the essential components needed to facilitate the actual reaction in the thermocycler under the right conditions to amplify the gene. And then once all of that is done, you need some sort of method to confirm that that has been successfully done. And we use gel electrophoresis, but there are other techniques to do that. So for the purpose of this webinar in general, the focus will be on STA1 exclusively, just because it's the one most people are interested in, as it affects diastatic yeast and their sugar consumption in beers as a contamination. So the following slides will discuss all of this in greater detail, but it will be pretty dense. So some background knowledge on PCR. It's a molecular technique used to amplify regions of DNA and was discovered in 1985 by Kerry Mullins, which is this guy on the side. Under the right conditions, you can replicate the DNA strands to be exact and confirm the presence of them once that's completed through a detection method like gel electrophoresis. And typically these are designed to do to target regions of the genome that do encode for genes rather than non-gene coding regions, because typically genes will make proteins and then those proteins actually perform some function we can measure later on. So DNA in general, or deoxyribonucleic acid, is found in all of cells, whether they are human, yeast, or bacterial. Everything contains DNA and it is stored within the nucleus of the cell. DNA itself is comprised of all of our genetic material stored as a code, and each person's code is a little bit unique and different, which makes us us. But all of this code is made up by 4 nucleotides. And those are adenine, thymine, guanine, and cytosine. They just repeat throughout, and that makes DNA. DNA also inherently contains a sugar phosphate backbone, which would be this blue ribbon in the drawing. That will come into play a little bit later on in terms of structure, but we'll talk about that later. So the 4 nucleotides, again, adenine, guanosine, thymine, and cytosine, together form all genetic codes. Throughout this presentation, they can be called nucleotides, DNTPs, or bases, but they can also, in literature and out in the world, be referred to as the building blocks of life. Adenine always pairs with thymine, and guanine always pairs with cytosine. These nucleotides give DNA a direction, and that direction for synthesizing DNA is 5' to 3'. Where the notation comes from, I'm truly not sure, but it's 5' to 3' for all intents and purposes. Each nucleotide contains a phosphate, a sugar, and a nitrogenous base. So these 3 components here. The phosphate will form a bond, a phosphodiester bond from one nucleotide to the free hydroxyl group of the sugar. So they kind of go in together, and that continues to build and connect until you have your gene. It makes more sense in this image where you can see our first nucleotide is adenine, and you have your 5' end, and then your sugar, and then a 5' and a sugar. And then at the end, you have a free 3' and a free 5'. So your DNA would be synthesized in a 5' to 3' direction. That's DNA extraction. Sort of the best way to think about it being read is you can only read a book in one direction. So you can only synthesize DNA to be an exact copy in one direction. If you do it backwards, it's not going to be the same as what you intend it to be. So this will lead us into actually extracting DNA from cells. And in order to do that and to obtain a purified DNA that you can use in your PCRs, you need to lyse open a cell, which just means kill it and break it up. There's 2 sort of methods you can use to do that. One is using chemical approaches or detergents, and one is using physical shearing, or basically just ripping it open with glass beads or any sort of physical instrument. Regardless of what you choose to do or what technique you use, the goal is to end up with free DNA, which is in solution, free of proteins, cell wall fragments, and debris. But when you break it up, it's all there, all in solution. You kind of have to sort through it. In my opinion, for all intents and purposes within the brewery, Instagene matrix from Bio-Rad is the easiest DNA extraction method. It does work for bacteria. You need to make a little bit of adjustments for that, but for yeast, it's extremely easy, very straightforward. And it's a pretty interesting technology. So it's comprised of 6% Chelix beads, which basically is just a bunch of beads in solution, and they go around and collect magnesium chloride, which inhibits PCR if it's too high. So we clean all the magnesium chloride up. You incubate your samples in the Instagene matrix at 56 degrees in a water bath, and this inactivates connective tissues in the cell wall and DNases. DNases are enzymes that degrade and break up DNA, and since we want DNA intact, we don't want to break it up, so we have to inactivate those. You then boil your samples to lyse them open, break them up, and denature all the proteins. The Chelix beads kind of continue to clean up any released magnesium chloride. You then spin the samples down to make a pellet, and DNA is soluble. It's the only thing that's left up in the supernatant, which we can use later into our PCR reactions. There are other methods and kits available out there, just as there is with pretty much everything. QIAGEN is a really great company. I like them a lot, just because they're super easy to use. They're fairly affordable, and yeah, they're basic. But extraction kits using a column purification system are more sensitive, meaning you can start with less genetic material. So for the Instagene one, we do everything based off of colony PCR for now, whereas the extraction kits would work using beer samples. And all column purification, whether it's QIAGEN or a different company, pretty much follow the same steps, which is a lysis step. So it's a chemical to break open the cells, a binding step, which allows DNA to bind the internal column. So we can follow this little schematic on the side. A wash step is next. It basically passes through the column, rinsing off any excess salts and proteins that might be, accidentally attached or floating around still. And then an elution step. So this final step is a chemical that has a higher affinity to bind the column. And it actually pushes all the DNA off the column. It binds the column, and you collect just pure DNA into a tube. So whichever protocol you choose, totally fine, as long as you're able to conduct the actual work at your brewery. Since they yield the same product, that's great, but you need to know what equipment you use. So before buying any of the kits, they all have PDF versions of the protocols online. Look them up. So Instagene, you have to store it in a fridge. You have to have a water bath that's stable at 56 degrees, but you can use benchtop minifuges and small centrifuges and small vortex to mix everything quickly. And there's no real long steps. Whereas for a lot of column extractions, they're long vortex steps, usually requiring bead tubes, which need an adapter for a vortexer. And you also have to centrifuge at really high speeds for long periods of time, which can burn out some motors if you don't have a proper equipment. Some need ice, which is also wild when you figure that out. So continuing on, regardless with what you pick, just make sure you research the method or the kit you buy beforehand. Regardless of which you choose, you'll end up with purified DNA that you can use in your PCR reaction tube. From this point on, we will be calling purified DNA template DNA. So a little bit of a change up in wording. But in order to get any sort of amplification from your PCR, you need to create master mix solution. And this will actually have all of your primers, nucleotides, and your polymerases, in your tube so that it can facilitate all the reactions. What the heck does all this stuff mean? This is where we're going to break it down into the components of the reaction. So the DNA template we've talked about, that's our purified DNA. And then we have to discuss primers, polymerases, DNTPs, and then buffers and cofactors, all of which are those master mix components. So our primers are specifically designed DNA molecules that are specific to our target region of our DNA. They bind a little bit upstream the gene just so that they can capture the entire gene, and they facilitate the reaction pretty much. So if you have your template DNA, it is double-stranded. Under certain conditions and temperatures, which we'll talk about in a minute, they unwind to become single-stranded, and then your primer pair will find their complementary strands, bind to the right area, and then it facilitates all the rest of the primers and nucleotides to complete the strand, and then we end up having complete copies. So each primer needs to be designed to flag or connect or anneal to their proper strand. And yeah, that's pretty much primers. DNA polymerases are special thermostable enzymes, and they transcribe or actually read your DNA. So they find the primers and synthesize the target gene by adding bases in a specific order following the genetic code. So basically, this whole process is kind of like building IKEA. You have, your set of instructions, which is your DNA template, your primer, which is your little pointer finger, and you start and you read all of your instructions, and you are the polymerase, and you add your certain DNTPs or your, pieces of the furniture you're building in a certain order to get your final template or your final DNA strand or your final wardrobe or whatever you're building. So the DNTPs get added by the polymerase in a certain order following a set of instructions that are illustrated on the DNA template or your genetic code. Buffers and cofactors, if this is still the same analogy with the IKEA, they would be the nails and bolts, certain pieces that are required to facilitate the actual PCR reaction and the actual building process. So for example, early on, we had to remove all the magnesium chloride from the DNA extraction process. And the reason we have to do that is magnesium ions have to be involved in the process, but in measurable amounts and in controlled amounts. So we actually have to add back magnesium chloride to increase and decrease the specificity of your DNA polymerase. Other essential components are also included, and this is potassium or tris hydrochloric buffer. The next couple of slides are a little bit beyond even me. I did not do biochemistry, but it's explaining how magnesium chloride works in the reactions. So magnesium ions specifically are needed to bind DNA polymerase's active site. So it's a site that allows the enzyme to actually facilitate the creation of those phosphodiester bonds, where you're adding your phosphate to your that extra free OH group on your sugar to facilitate the actual connection of the nucleotides. And it also facilitates the binding of your primers to your DNA template by stabilizing negative charges on the phosphate backbone. Yeah. Typical concentrations of magnesium in the PCR master mix should be about 1 to 4 micromolar. If you have too low of a magnesium concentration, you result in little or no PCR product. Not enough is binding, not enough can be produced. And if you have too much magnesium, you end up getting the primers binding absolutely everything, and you get a whole bunch of non-specific product that are the wrong size and just codes for nothing. So they're binding too much and just kind of making a mess. You still need potassium ions. They help with the magnesium ions stabilize the phosphate backbone and the primer annealing. Typically, most master mixes will have some concentration of both, but for the majority, it's either you have a magnesium-based or a potassium-based master mix, and then there's trace amounts of the other ion in there just to help facilitate stabilization. That's the hardest, most complicated part of this, so we're done. The components of a PCR reaction continued. All of this needs to be incorporated in the right amounts into your reaction tubes with your DNA template to facilitate a proper protocol. So, tweaking this protocol can be really difficult. You need to make sure you have the right concentration of magnesium chloride, you have the right concentration of primers, you have the right amount of your DNA template, your annealing temperatures are right. So it can be a lot to try to make a PCR protocol from scratch, which is why it's really good if you can source it from a paper and someone else has maybe done it. But all protocols for PCR pretty much follow the same 3 basic steps or 3 basic cycling processes. And the 3 steps are denaturing, annealing, and extension. So when you denature, it's when you unwind the double helix of the DNA into single strands so that your primers can bind. Annealing is when the primers actually bind to that DNA, which signals for the polymerase to come. And then your extension step is where your polymerase goes along and reads the DNA to actually produce and incorporate those nucleotides to make a new template strand. This step is then repeated for 25 to 40 cycles, hence PCR polymerase chain reaction. It ends up turning into this huge chain reaction, which we'll talk about in a second. But to go through the steps in a little bit more detail, your denaturing step typically occurs at 94 degrees, but this is based on the DNA polymerase that's used in your master mix. So at 94 degrees, you will unwind your DNA. This allows the polymerase and primers to bind, and you end up with 2 single strands. And prior to the actual cycling, where the steps are repeated, you would want to have an initial step of denaturing, so that all of your initial template DNA is denatured to start the process. You go into annealing, where your primers actually bind. Each primer pair has an optimal annealing temperature, which should be 5 degrees lower than the melting temperature that's stated when you order the primer. And it should be 5 degrees lower than your lowest primer pair. So if primer 1 has a melting temperature of 60 and your second primer has a melting temperature of 55, your optimal annealing temperature that you'll include in your PCR protocol would be 50. So it's 5 degrees lower than your lowest primer, which is 55 degrees. So an annealing temp of 50. Annealing temperatures, again, are kind of dependent on your Taq polymerase and on your primers, based on what you're using and what your range for those are. But they typically are between 49 to 59 degrees Celsius. So the last step is elongation or extension. During this step, DNA polymerase is directed to the primer's location, actually attaches, and then reads or transcribes the DNA template to create a new complementary strand. As you can see, when it's all done, you end up with 2 double-stranded DNA again, like two copies of double-stranded DNA, and this is when the process will repeat. This elongation step typically happens at 72 degrees. Again, this is optimal for Taq polymerase, which is just the most common polymerase that is used in most applications. And again, it reads always in 5' to 3', so your primers will always bind on the 3' end to create a 5' end so that the polymerase will read 5' to 3'. So in both cases, its complementary strand is the 3', the 5' is then created with your primer, and then it will read it along. And why it's called chain reaction is because you start off with, 1 copy of your DNA. After 1 cycling has gone through of all 3 steps, you end up with 4. So 2 copies of single-stranded, and then 4 copies of single-stranded, then 8, then 16. And after 40, it exponentially just goes through the roof, and you are able to later detect it. Something to note, though, is if your DNA template is missing the gene that you're looking for because it just doesn't exist, none of this happens. It just goes through the cycles and nothing will occur. So gel electrophoresis, we're going to talk about that because it's what we use mostly at Escarpment Labs, and this is our detection method. It's also one of the most common detection methods. But anyways, once the reactions are completed, you can test to see if it was successful. You do that using gel electrophoresis. It's a yes or no detection method. The presence of a band on your gel indicates successful amplification of the target gene. So an example would be if you're testing for Spooky Saison from Escarpment Labs, which is diastatic, which does have STA1, and Vermont Ale, which is non-diastatic, does not have STA1. For that STA1 gene, you would expect that amplification would work. You would see a band for your Spooky Saison, but you would not see anything for Vermont because it doesn't actually contain that gene. So the gel electrophoresis is kind of cool. It's a cool little idea. It's used to visualize endpoint PCR. So if you hear that term, it just means people aren't doing qPCR. They're running a normal PCR and then have some sort of detection method afterwards. I have one slide on qPCR because I figured somebody would ask about it. But we don't use qPCR. We just do regular PCR and then gel electrophoresis. Gel electrophoresis involves making a gel, and then you would lay your gel in this apparatus with a buffer, and then you load all of your DNA into wells, and then it creates an electrical current, and your DNA runs down the gel. Advancements in the field has gotten pretty cool. I want one of these machines for Escarpment Labs. It's an Azure biosystem. Basically, it makes the gel for you in real time. So you don't have to wait till the PCR is done to run your gel. It just populates it onto the screen and you can print it. So that'd be pretty fun, Richard. qPCR quickly. So it actually stands for real-time PCR, and it works by monitoring the progression of the amplification in real time. And this is based on fluorescence. So it basically takes a screenshot of all fluorescence at the beginning of the cycle, and then at the end of the cycle, and then the beginning and the end, and it compares them, and then it measures through an algorithm how much amplification has gone from your initial amount of template to your current amount of amplified DNA. And then it eventually shoots up if you do have it once it hits the exponential phase. And then you can calculate the exact amount of DNA or amount of starting material you had if it was a high contamination or a low contamination based on the curve. So we don't do qPCR pretty much only for the reason that, other than it's expensive, that for us, any level of contamination is just unacceptable for our products. So if we notice any contamination through endpoint, it doesn't really matter what the threshold level of that is. It's just we're done. Like we have to start over and make something new. So if it's a contamination where it shouldn't be. So that's the only reason we don't use qPCR. There's nothing wrong with it. It's pretty great. You don't have to wait for the gel or for the PCR reactions in the thermocycler to actually finish because you can just watch it in real time as it goes along. So it's kind of cool. But back to gel electrophoresis because it's what we use. So it works based off of charge. I mentioned earlier the sugar phosphate backbone is negatively charged. So that gives DNA inherently in total a negative charge. So when you load your DNA into wells on the negative side or the negative anode and then run the electrical current, it will run to the positive anode and it will separate out all your bands on the gel, which you can detect then. DNA is actually separated by size, so any small fragments can run faster through the gel and will end up closer to the bottom, and any larger bands or larger sized fragments will end up at the top. And when you run this gel, your first lane is always with a commercial ladder that you purchase. When you buy it, it comes with a printout and it says, you know, this band is 1,000, this band is 800, this band is 500, this band is 200. They're always the same. And you can pair up where your gene or your amplified amplicon ends up. So for example, STA1 is about 800 base pairs long. It's like 830. So when you run it, you would want to see if you can line up that 800 base pair with the size of your fragment to confirm that you actually amplified STA1 properly. Traditionally, you would add ethidium bromide into the gel itself when you cast it or you make it, and then you would detect it under UV light. There's a lot of problems with that for human health. Ethidium bromide is a mutagen. So the way it works is it actually sort of just like embeds itself inside a double-stranded DNA. And if it can do that on a gel, it can do that to you handling it. So there's huge handling restrictions and stuff when using this stuff. You need to have special cabinets. Luckily, nowadays we have much safer stains and dyes, and we don't have to use UV light all the time, which is another health hazard. So it's come a long way, and it's a lot safer. So this is an example of a gel that I have run testing STA1, the STA1 gene on all of our diastatic strains. It looks like a lot. So this is all of our strains. The bottom half of each gel is me testing for another gene called ITS. ITS is found in all yeast cells. So I use this as a positive control to confirm that all of the DNA extractions work and that I can actually amplify a gene in that DNA that all the DNA should have. So I try to make sure that it's in everything except for the last lanes here, which are all my negative controls. So instead of adding actual yeast genetic material or template DNA, I add water, and then that won't amplify at all. There's no genetic source for it to amplify, so we get nothing. And then the top bands are all our STA1 experiments to see if they're STA1 positive. And based on everything else, we can confirm through all of our controls that they are indeed true. And through this, we were able to actually determine that our Saison Maison, which we recently released, is not diastatic. So that was kind of fun, and that led to the commercial launch of that strain. So it was a fun little experiment. But yeah, so this is what we do a lot internally. We have a lot of controls with our system, and we use electrophoresis, and it works totally fine for what we need. So how do you run your own ST1 experiment using existing equipment that would look like that? You do have to buy some stuff. You have to get some additional equipment. But we still use a lot of the equipment we originally purchased for the Invisible Sentinel system. So that's our SimplyAmp thermocycler that comes with it, the centrifuge that comes with it, and then a little desktop vortex that we bought as well to use for it. So we don't personally use the Invisible Sentinel extraction tubes. We use the Instagene. However, it would totally be fine. You can totally use those if you want to. They work well. They purify DNA. They do what they say they do. So you can keep that as well if you still plan on doing a different version of the detection method, which is what we're kind of suggesting. Additional materials you would have to buy we buy all of ours through Mini PCR. They're very affordable. We really like the company. So we buy our master mix ladders, nuclease-free water, and this gel electrophoresis system from them. The electrophoresis system, I think, is like $300. It's not horribly expensive. And then a master mix that'll run 200 reactions is about $25. And then each of the ladders, which can be used for so many reactions, I think is like $10 to $20, depending on the ladder. So we also purchase Instagene for our DNA extractions. So our actual protocol that we do, it's a little difficult to show it on this, so I tried, but If you do need to see the full protocol, it's also on our blog posts. But we use colonies from plates, put that into a sterile microfuge tube aseptically with 200 microliters of the Instagene. Follow the protocol, which I mentioned earlier, but it's vortexing it, letting it sit in a 56-degree water bath for half an hour, boil the sample for 9 minutes, and then centrifuge it down for 3 minutes at 12,000 RPM, and then use the supernatant in the downstream PCR reaction tubes, or the downstream PCRs. To actually set up our master mix, we buy the commercial master mix. So this green stuff, it has everything in it. So it has our polymerases, our DNTPs, our buffers and cofactors. All we really have to add is some water to dilute it to the working concentration. And we have to add the primers for whatever we're testing. So if I was testing for ITS, I would add the ITS primers. If I'm testing for STA1, I add the STA1 primers. In this concentration, so 9 microliters of sterile nuclease-free water, 5 microliters of their commercial master mix, and then 0.5 microliters of each primer into each of these tubes. It's really hard to pipette that small of a volume, so we actually make an aliquot. So if we're testing 8 samples, we'll multiply that recipe out by 8 or 10, make one big solution, mix it up really well, and then put it into each tube. That's also really great because it ensures that all the tubes are consistent and the same. They have the same ratios, so if one fails, they should all fail. Whereas if you're doing it in individual tubes, you open yourself up to way bigger pipetting errors and missing one of the primers or something. So we create our master mix, and then we add 10 microliters of our DNA template into each tube. We run it through our thermocycler for our protocol. We actually adapted a protocol from a paper, which I believe Richard has access to and can post in the discussion, hopefully. But the protocol is also on our blog. It's right here. We initially denature or unwind all of our DNA at 94 degrees Celsius for 15 minutes. Then we run 30 cycles of denaturing, annealing, and extension. Our denaturing is at, again, 94 degrees for 1 minute, annealing at 49 degrees for 2 minutes, and then extension at 72 degrees for 2 minutes. We do a final extension step. If you have an initial denaturing step, you should have a final extension step. Kind of lets everything just finish up if it wasn't completed for 10 minutes at 72 degrees. And then we hold our samples at 4 degrees Celsius. Just cools them down and we can handle them. To detect it, we use the gel electrophoresis system from PCR. We follow their instructions to make a 2% agarose gel using all the reagents. They use an ethidium bromide replacement, which is much safer. The SDS says it's completely non-toxic. We still handle it very well, like make sure we have all the appropriate PPE, just because if ethidium bromide is super bad, whatever replaces it is probably super bad. But they said it's safe. I trust them, but I still take precautions. And then they use blue light, not UV light, which is also safer to use. And then you end up just taking a picture of the final gel with your cell phone. So all the pictures I showed earlier of our experiments are with a cell phone. So I don't want to really harp on the invisible sentinel system. That's not my intention here. I'm just trying to offer a different solution to use their system with a different detection method that is cheaper and just provides more information than the little cassettes that they use. You can use a lot of their stuff. I would say you can use all of it up until the detection method. So for example, you can take your beer sample, follow their enrichment steps, and then perform your DNA extractions. And then either use your internal protocols and your own master mix for your STA1 or whatever you're testing protocols, or follow it through to use their master mix and their PCR protocols, and then visualize it using gel electrophoresis. Their kits are very costly. We found them very expensive, especially when you're trying to build in some sort of controls. Ours, like our internal protocol that I just shared with everyone, costs us $6 in materials to run one sample. So you can develop and adapt your own PCR protocols as needed based on your budget and your comfort level. So this leads us to our take-home message. If anyone has watched any of our other webinars, we preach to the highest whatever about plating. So one of our take homes is if you are doing your beer enrichments with the invisible sentinel system in the EB broth, it takes 2 days before you can actually run your PCR if you follow their protocols. In that time, if you plate your samples onto WN, LCSM, MRS, or any other sort of plates, you get so much more information than just a cloudy tube with some growth. You can find out if your yeast has cross-contamination, if your yeast is healthy, if you have bacteria. You can see if that yeast is growing on LCSM and when it shouldn't be, and that would be a clear indication pretty quickly that you might have a diastatic contamination, since they correlate really well. And all of this is on the same timeline as that actual beer enrichment step. Something else to note is that DNA is not alive. The very first step of our PCR was to kill everything and take the DNA from it. So, yeah, I mean, say you had a tank that had French Saison in it previously, you put in a new batch of beer or it comes in contact with it, and then you boil it and then cool down, do everything, and then pitch Cali, and you run your PCR test on that. That dead French Saison 100% would have left some sort of genetic material in that tank. If you get a positive result from your PCR, it's probably true, but you were just picking up genetic material from French Saison, not the Cali. So plating will actually determine if the contamination that you're detecting is active or not, if it's actually alive, or if you're just getting some sort of residual amplification. With our protocol, as I mentioned a little bit, we can build in a lot of controls. So we built in all those controls I talked about earlier. So positive controls, negative controls, controls to determine that our DNA is amplifiable. And you can't really do that with the invisible sentinel system. And if you do that, it will cost you so much money. But you can really only run a positive and negative control because they only really detect for one thing. But still, even if you add those controls, it's a lot of money. And we find that the actual cassettes can be a little bit ambiguous sometimes, especially because each one is individually packaged. Each one could be subjected to its own sort of internal defects. There's no COA guaranteeing that the test is going to work accurately when you use it. So there is a lot of variation in just inherently in the way that the system runs. Yeah, again, I'm not trying to like harp on Invisible Sentinel. It's actually a pretty interesting technology. PCR in general is just really complicated. It requires a lot of setup, and it's hard. Like it's hard to build in controls and to validate and confirm all of your results that you're trying to obtain. At the end of the day, there is no right or wrong PCR system, but you just need to use a system or a protocol that you can afford, that you can trust with whatever information it tells you, and that you're actually comfortable using at your skill level. My personal take-home message is to never run a test that you cannot use to make a decision. If you cannot trust your results to actually act on them, there's no point in running that test. And if you ever run a test, you need to keep records and write it down and have it somewhere accessible for future use. For us at Escarpment Labs, we need to trust all of our results so that we can actually communicate those to our customers with confidence and actually stand behind them. So this is why we have invested so much time and effort to develop and validate our own protocols. So before all of the world went crazy and hornets started coming at us, we were working on developing a one-day beer DNA extraction test, likely using those column purification DNA extraction methods and PCR protocols to test for STA1 for brewers, using all the controls shown previously and adapting existing equipment you may have purchased for other kits. Yeah, we're going to try and validate that. It would be super useful to get a one-day test from beer to gel, I guess, and have it actually be super reliable with a whole bunch of information you can use. Our goal is to have this by December 2020, if everything works well, so a little bit of a Christmas present. And thanks for listening. Thanks for dealing with those technical issues earlier on. Stay safe, stay tuned, and we will discuss questions and stuff. If I can figure out how to stop sharing. Oh, you did it for me. Thank you.
I did it for you.
Holy.
Thanks, Alex, for the informative presentation. I think it's good to just have a bit of an overview of PCR, understand a little bit better about how it works, and then, taking some of that fundamental knowledge and understanding how it may be possible for brewers to, start taking a little bit more of a DIY approach with this stuff. I think that's sort of, the central thesis you were getting at is that you don't have to buy regardless of where you're buying them, you don't have to buy the expensive kits. It is possible to do it yourself. And there are people that are, interested in helping out with, getting breweries up and running with PCR, because, at the end of the day, the more breweries that are up and running doing this kind of testing, the better we have, in terms of our quality landscape across the board. So that's always exciting to us when we hear breweries looking to step up their quality control game.
Yeah, no, for sure. It sounds super scary, and I know I was just spitting out words like rapid fire, and it sounds really overwhelming to get your head around it. But once you start actually doing it and doing it hands-on and kind of playing with it, it's way easier than it comes across. But you're right, it takes a lot to actually develop the protocol and try to get that up and running. So hopefully
I'm having PTSD flashbacks of having flown to Germany for a summer internship and meeting the professor, and my first day, him handing me some materials and saying, "Okay, go do a PCR. I need to know. I need to see what you know."
Well, that's what you did to me. That's how I got my start in the lab. You're like, "Here you go," and I was like, "All right." Took me.
Yeah, it's the school of hard knocks, old school microbiology, all the way.
Yeah.
That's awesome. We do have some questions, which is great. I'm gonna start from not a lot of votes, but I'm gonna start from the top question. The question is, what is the best way to integrate PCR into a QA program alongside plates? Should you check every batch or only use it as a secondary method for testing after having a positive result? By that point, would it be too late?
Up to you. So, if you're incorporating it with plating, you have to at least wait 3 days for that stuff to grow. Sometimes you get information from your plates. So if you're growing French Saison on your plate and it's LCSM positive, that's totally expected. If you're growing Cali and then you see it's not expected, you would want to test that. We test. So we initially plated all of our strains out. We got to know our strains and what grew on what, documented it. So if we see something that's an anomaly or unexpected, then we go ahead and test it. And that's kind of what I would recommend as well. It is a lot of money to just kind of do it all the time. It's also a lot of work to do it all the time. But I would say, at least to start, do it if you see a contamination on something that you're not expecting. That's how I would initially incorporate it, I think. There was a second half to that question, but I don't really remember.
Would you use it as secondary testing after having a positive plating result?
Yeah, I would do secondary testing, I think, for that.
I think, I think this is something we've talked about in other webinars as well. Is I think it's really good to have a plate, because the plate gives you so much information. If something does grow, there's a lot of these secondary tests you can do, and you covered a little bit of that as well. Like if you have a yeast or a bacteria and you have a colony on a plate, you can look at it under the microscope.
Yeah.
You can do some of these basic secondary tests like the catalase test and oxidase test. You can get a lot of information that you might not be able to get out of a PCR. PCR is it's very, very laser focused, right? So, in my view, plating can cast a wider net, and then PCR allows you to really narrow in and identify this, the specifics of whatever it is you've found. That being said, of course, then taking that approach does slow things down, right? Like there is definitely an argument to be made for doing both, at the point of sample collection and then being able to compare the results as well, versus using PCR as a secondary approach. It kind of just depends on, a lot of it depends on the structure of the rest of the business, right? Is that beer in a distribution network for a little while where, if you have to wait an extra 3 days, you might be able to pull it, or is it going out to stores or customers right away, right? Where then you have no control over the product, and that might depend on how the QC program's set up.
Yeah, you can build that into your QA system though if you're testing everything a little bit ahead of time, knowing what your timelines for those are.
Yeah.
What I will say too is if you follow our protocol and you have a colony on the plate, you can have results. If you start it in the morning, you can have it by noon. Like our protocol is only 3.5 hours for the cycling, plus half an hour on either end to set it up. So it's like a 4.5 hour thing. So you could have results same day as the day your plating is done. If you're diligent, you time that or plan that into your QC regime or QA regime.
Yeah. And that is one of the benefits of some of the qPCR, real-time PCR systems out there, is that that you can get, some of them are done in an hour or less, right? So you can get results even faster with those. So there is an advantage to those in terms of timing and not having to run gels and stuff like that. I've got another question. I'm curious to know if you know the answer to this. Does the polymerase denature during the melting step? If so, is more polymerase needed to be added between each cycle?
I don't think it denatures. I think that's why they use Taq polymerase. It's heat stable, so it denature. I think what might happen is the active site or something gets reconfirmed during the or changes its conformation or something, which does something. But yeah, no, I'm going off on a tangent here. My answer is no. I don't think it actually denatures, but it could.
Yeah, you're absolutely right. That's why that enzyme, the Taq polymerase, was such a major discovery, because PCR was developed using DNA polymerases from E. coli, so it was not nearly as temperature stable. So there were issues where it may have had to be added in, and obviously back in the day they're not doing this with a fancy machine. They're taking a tray from one water bath to another one to another one.
Yep.
Doing PCR uphill both ways. So yeah, the discovery of this this temperature stable, heat stable polymerase from this hot spring bacteria called Thermus aquaticus, so TAQ, was a revolution, because it meant that you had this enzyme that you could heat up to 95 Celsius and it cool it back down and it still functions perfectly.
Yep.
So that's pretty awesome.
Cool.
It means that you don't need to add it in after the fact. You put all your stuff in the tube and throw the tube in the machine, and away it goes.
Cool. Glad I got it. I used to know all this just like that, but it was a lot of a refresher for me, having to actually dive in and research things that I learned in first and second year university, which was a few years ago now. Having to actively look up things again, I was like, this is insane. So
yeah. We have one final question, unless anyone else would like to ask anything else. Not sure exactly how to answer this one. The question is, could one also use minimal media on plates to test for STA1?
Minimal media, is that like those Petri film plates, or what's a minimal media?
Well, I mean, usually when you see that's referring to an agar medium that has basically the minimum
requirements
nutrients required for a given microbe to grow. So it would be used it's used for selection in some ways. For example, if we're looking at yeast, you can select against mutants that are deficient in amino acid synthesis by plating on minimal media, for example. Say for example, when we're making if we're making hybrids, those hybrids are no longer deficient in amino acid production, so they would grow on a minimal medium. I'm not sure if that would work, in terms of STA1, because at the end of the day, the difference in those is function of the gene, and not and that doesn't necessarily have as much to do with nutrients. I know that sort of this idea may have been tested in the past with trying to select for diastaticus on starch-containing agar, but something we talked about in our Diastaticus 101 webinar was that that works, but it's really slow. It can take 3 weeks for some of these things to grow on agar. I think you did some of that stuff as well, Alex.
Yeah, Iz did all the plating, and then I did all the PCR end on the same strains, and then we compared all of our results to see what matched up and what didn't, and it was really good. But PCR, I was done my PCRs in like 2 days with all of our strains, and it took like 3 or 4 weeks just to see any sort of growth on any of those plates, which was crazy.
Yeah, so that's still,
see if anything was happening. It was like insane. So
that's still very much like a holy grail with diastaticus is a some kind of culture-based medium that directly detects the gene function and is fast. Thus far, nothing has seemed to work, and I know a lot of people I know like Omega Labs put a lot of energy into trying to develop something as well, and they were able to improve LCSM, but at the end of the day, that's still not direct detection of the STA1 function. So it's still a bit of a mystery.
Yeah, it would be great. I mean, maybe one day we'll take a crack at it if we have an eager student who wants to do that. But it would be.
I've got another crazy idea, but we'll talk about this later.
Offline. Sounds good.
Okay.
Cool. So I hope that answers.
I hope so. We have one more question, and I think we'll wrap it up after this. The question was, how limiting are commercial primers when screening for bacterial contaminants? And are there any custom primers available?
So we use like a universal primer. Universal. It's kind of similar to that ITS one I mentioned where it's found in all yeasts. We use one that's found in all bacteria. Use it to identify it later if we send it for sequencing, what that bacteria is. So it's called V6R. I'm not sure what the actual name of the gene is, but that's the name of the primers. It's V6R and V2. There is another one. It's called NL1 and it might just be NL1, but it's another gene, does the exact same sort of thing. Bacteria all have it. So you can target genes for that. And again, those commercial primers, we found them in papers. Those are things that we have done, but I will tell you that optimizing those protocols to try to get it to work for all bacteria is very difficult. It took me 8 months to get our V6R protocol working up and running completely for it to be able to widely screen everything that was going on. And that was 3 to 4 PCRs a week, literally for 8 months. So it's a lot of work. You can find it, but again, there's nothing ever that's like totally perfect. There's always something that breaks the rules.
Yeah, that was a funny one. We had some, just to provide some context, we had some lactobacillus strains that seemed to be impervious to some of the common lysis techniques for PCR, and it was really tough to get good DNA out of them. So that made it hard to PCR them and actually find out what the heck they were, which was kind of scary in a way. But yeah, that just speaks to the enormous diversity of bacteria. I think the question might also be about some of the commercial kits that are available and how limiting those are. And I will say that if you have a kit for lactobacillus, it's probably screening for hop resistance gene, which might mean that it's not actually picking up all lactobacillus. And given that the hop resistant genes jump around between bacteria on sort of little genetic islands called plasmids, even a non-hop resistant lacto can pick it up later on. So I would say it's still a risk. I would, it's always worth talking to those suppliers and understanding exactly what those kits are testing for. And yeah, we have this huge body of scientific literature that, luckily, anytime anyone studies anything with PCR, they have to, list the primers they've used and the conditions they've used, whether it's reproducible or not. That information's there, and it's really cool that the technology to synthesize, little fragments of DNA is so cheap and fast that, we order from a company called IDT, Integrated DNA Technologies, and there's other ones as well where you can go in, copy and paste your primer sequence, hit go, and it shows up in a couple days. And it's like $12 a primer. So it's very, very affordable. And that's enough for, a couple hundred reactions.
Yeah.
So,
More,
brave new world.
Out of it.
Yeah. And yeah, like, again, you as a anyone as an you know, an amateur or as, you know, as a lab professional, you can design your own primers too if there's something interesting that you want to screen for, right? You know, even beyond STA1, there's other interesting genes out there or interesting organisms. You can just find, you know, that gene and copy and paste that, and there, even online you can search like PCR primer generator, and you can get software to generate primers for you and order your custom primers and test them out. Obviously, like, primer design itself is a nuanced art. I know there were some issues down in the US recently related to qPCR primer design that caused some problems. But at the end of the day, it's actually a perfect problem for, you know, for software to address, because we understand a lot of the, you know, physics and chemistry of primers binding to DNA and helping to amplify them. So it's a pretty well understood thing, and you know, we're able to, you know, predict and generate primers fairly easily. Like, we had experimented with this idea of, you know, can we find some unique DNA and kvike so that we can make a, you know, is this kvike or not primer set, and that never really worked out. But, you know, these are the kinds of ideas that you can explore.
Some fun project Jonah and I were going to start up was I think we ordered the primers, or we were designing the primers to test for female specific weed plants. See if we could grind up some weed and see if we could test to see if that plant was female or not. So you can do whatever you want, right? Like, it's a little bit of the Wild West if you have some sort of knowledge and can use some of the free accessible tools around that can help you with your process. So yeah, we were going to play around with some fun stuff. And it is fun. PCR can be fun. It can. It's not fun when you're doing it for 8 months straight on the same thing, but it can be fun. It's like, it's really interesting, and it is an interesting tool to play around with if you have it at your disposal.
Absolutely, yeah, and it's, you know, really helping us all over the world solve all sorts of problems and you know, get to the bottom of biological mysteries faster. All right, I think that's it when it comes to Q&A. So I just want to say thanks again, Alex, and for anyone who's watching, I just want to mention we do have a brewer interview this Thursday with Avery Swanson from Keeping Together Brewing. And we'll also be talking about good wild yeast hunting on Tuesday as well with Jonah from our team. If you ever want to learn how to catch your own wild yeast and test them out, and we really find some truly unique local flavors, we'll try to help, you know, answer those questions and make it easy, because, you know, it's not just leaving a mason jar of wort out and hoping for the best. There's ways to really increase your chances of success and make it worth your while. So thank you, everyone, and have a good night, and we'll see you soon.
Thanks, everybody.
Thanks, Alex.